Welcome to Structure Club. I'm Scott Stagg, a professor at Florida State University, where I use cryo-EM to determine the molecular mechanisms of membrane remodeling.
And I am Ashwant Francis, an assistant professor at Florida State University. My lab studies structural biology of viruses and how they infect cells. Together, Scott and I, we created this Structure Club as a journal club podcast and YouTube channel where the papers are given by the authors themselves.
Today's speaker is Guilia Zanetti from the Francis Crick Institute and the Birkbeck University in London.
Welcome, Julia.
Thank you.
Julia got her PhD in 2009 in Oxford with Stephen Fuller. She then moved on to do a postdoc with Randy Sheckman at Berkeley, and she finished that around 2013. And then she moved on to her own independent career and started her own lab at the Institute of Structural and Molecular Biology at Birkbeck College in 2014. And now she has a secondary appointment at the Francis Crick Institute that will run until 2031. In her own independent research, she has been focused on the molecular mechanisms of membrane trafficking, particularly with COP2. And she's going to tell us about her paper, Multiscale Molecular Imaging of Human Cells Reveals COP1 and COP2 Vesicles at ER Exit Sites. That manuscript is currently under review and can be found on bioRxives. Julia, the mic is yours.
Thank you very much for the kind introduction, Scott, and thank you both of you for inviting me to give this Structure Club. I'm quite excited, so let me share my screen. Can you see my PowerPoint?
Yes.
Perfect. Okay, so, and a laser pointer. Now we're all set. Good, so thank you so much again. I'm really excited to be presenting our preprint, which has been out there since the summer. Hopefully, I'll be reviewed soon. And I just want to start with a disclaimer, which is that you're not going to see many structures in this paper. But I also want to start by mentioning that the work I'm presenting here has been a great collaboration with John Audia's lab in Wisconsin. John and I are both interested in molecular mechanisms underlying membrane trafficking in the early secretory pathway. And we came together to tackle the outstanding question of how membranes are organized at mammalian ER exit sites. So John's lab has got a great cell biology and fluorescence microscopy expertise and toolkit, while we are experts in cryo-electron tomography. So we joined forces to obtain a multi-scale molecular view of the early secretory pathway compartments in human cells. So I will start with a brief introduction and a summary of the outstanding questions before I dive into the paper figure by figure. So about 1 in 3 proteins in eukaryotic cells is translocated into the endoplasmic reticulum upon synthesis. And from there, it's transported to the Golgi apparatus from which it's sorted to its final destination. So this is called the secretory pathway, and cargo of the secretory pathway include most membrane proteins and secreted proteins such as hormones, antibodies, extracellular matrix components. So it's a fundamental pathway in eukaryotic cells. So these images here are from a review we recently wrote, and they summarize what we call the classical vesicular transport model for ER to Golgi trafficking in animals. So according to this model, small cargo-loaded vesicles form at a region of the ER called the ER exit sites. And these are formed by the membrane remodeling and concentrative action of a coat protein complex 2, or COP2. So before reaching the Golgi, the cargo is actually trafficked through an ER Golgi intermediate compartment, or ERGIC. This is in mammalian cells. And while we do not fully understand how ERGIC actually functions, we do know that it can act as a sorting station for recycling of cargo receptors and escaped ER resident proteins to retrieve them to the ER. A function that is normally performed in the cis Golgi in yeast, plants, and invertebrates. And the recycling activity back to the ER is attributed to the retrograde coat protein complex 1 or COP1. Right, so going back to COP2, we understand quite a bit of how it works, how it forms vesicles. This is from biochemical reconstitution and structural work, including some really seminal work on cryo-EM structures solved by our very own Scott Stagg here a few years ago. So in brief, the way it works is the coat assembly starts when a small GTPase, SAR1, exchanges its nucleotide and its GTP-bound form. It binds to the ER membrane and recruits the inner layer of the coat. And these in turn, serves to specifically concentrate cargo and cargo adapters into the forming coated membrane, and at the same time provides a structural scaffold that is necessary for recruitment of the outer layer of the coat, here in red. So here we have these rod-shaped complexes that assemble to form cages that stabilize the coated vesicles. While the timing of the coating and especially of the uncoating is not known, we do know that coat formation and uncoating are highly dynamic processes because the proteins from the inner and the outer coat themselves directly have GTPase activating function, basically meaning that as soon as the coat starts assembling, it also starts to disassemble. Right, so that's kind of the summary of the classical model as we know it. Despite the very strong biochemical and in vitro evidence for this model, in recent years, doubts have arisen that this is not the main or the only way that ER export can happen in mammals. So this comes from technical improvements in fluorescent microscopy, block scanning, SEM, suggesting that ER exit sites in mammals are actually not composed of coated vesicles, but rather by tunnels or direct connections between the ER and the ergic, where COP2 acts as a collar rather than a coat. And these alternative models are quite suggestive because the classic vesicular transport model doesn't really explain how large and abundant cargoes, such as collagens, one of the main components of our extracellular matrix, are transported. So that's the end of the introduction. Basically, in this paper, we set about determining how ER exit sites membranes are organized at the molecular level in mammalian cells using a multi-scale approach. This includes cryocorrelative light and electron microscopy and other techniques. So the idea here is to use a focused ion beam to mill cells to create a thin electron transparent lamella through the cells, and to use cryofluorescence to target a labeled object of interest. I'll come back to this. And then use cryo-electron tomography to obtain high-resolution three-dimensional reconstruction of the underlying cell volume at the fluorescent sites. Okay, so this is a very general overview of the workflow. This is the first figure. This is supplementary figure 1 in the paper, which basically validates our system. So here in panel A, you can see, so we chose retinal pigmental epithelial cells. These are a human cell line to perform this work. And you can see in these images, this is just immunostaining of the wild type cells that shows the distribution of ER exit sites as marked by one of the COP2 components, SEC31. We can quantify this. And we created with John Audia a cell line that has a knock-in for a halo tagged version of SEC23. This is another one of the COP2 components. And so this is expressed at the endogenous level. And here in this figure, we show that the pattern of puncta of the halo SEC23 is analogous to the wild type ER exit sites, and it does colocalize with SEC31, showing that this tag doesn't seem to disrupt the function and the localization of these COP2 components. Here we see some cryofluorescence images of these cells. So this is performed at room temperature. Here we are at cryo temperatures. And so all of these smaller black puncta are the SEC23. You can see them better in this inset here. This is before we mill the cells. And after we go and mill lamellas through the cells, we can collect again cryofluorescence images. And here you can see that we're left with a few puncta on the lamella. Of course, most of the fluorescence has been ablated away by the Fib procedure, but some fluorescence is retained. So the idea is now to go and target these fluorescent sites for cryotomography data collection. And this is how things would work in an ideal world, and that's how things work in our mind, according to our original plan, which we had to modify a little bit, because unfortunately, the correlation between the fluorescence signal and the TEM map is not accurate enough for what we wanted to achieve for such small structures as the ER exit sites. So here you can see in panel A, these are just two examples. So in the first panel here, we can see the cryofluorescence image of a lamella. So here is the support at the side, and this is the lamella here. And this is the equivalent TEM image of the same lamella. So if we are looking at collecting tomograms around one of these puncta, we would be focusing in this little red area there. So you can see that if our correlation is off even just by one fluorescent pixel, we are collecting in a totally wrong place. So it turned out that we couldn't really get a very high success rate using a direct correlation from the cryofluorescence to the high magnification tomogram that we need to actually see the molecular details of the proteins in our ER exit sites. So we added a intermediate low magnification tomogram step. So here for the technically minded people, we are using a total of 8 electrons over the whole tilt series and at the focus of minus 50 microns. So this basically gives us very high contrast, low magnification landscape that shows us where the membranes are. And so in 3D. And so from these reconstructions, we can actually see, these small black dots are actually ribosomes, so we can recognize the ER membranes, and then we can pinpoint where we have some sort of budding profiles coming from the ER, and then we can focus there to collect our high magnification tomogram.
Julia, just a
Yeah,
quick question. So,
sure.
so this intermediate level targeting, it's just your eyes, right? This is you saying, all right, I recognize ER and rough ER, and here's vesicles, so that's where I'm going.
Yes,
Okay.
exactly. This is because our if we just take the 2D projections, as you can see, this is like a
Mhm.
search map, a 2D projections. You can see mitochondria, you can see like the nuclear membrane, you can see some of the cellular structures, but these small clusters of vesicles are really, really hard to
Mhm.
recognize because they get buried in the projection. And so we need to do these 3D, and basically somewhere in this big box, we will have the signal that comes from the fluorescent puncta. And so we use our understanding of what it should look like to go there, which of course introduces a bias in our methodology because we are looking for a certain pattern.
But you
Um,
gotta do what you gotta do, right? So, yeah,
yes,
Yeah.
yeah.
absolutely. And well, I'll come back to that. Right, so let me move on to this is actual figure 1 from the actual paper. Those were kind of method Z supplementary figures. So here we have a couple of low magnification tomograms and a high magnification tomogram collected in the boxed area here. So this is actually turned out to be quite useful to collect the low magnification tomogram because, when we have the high magnification tomogram, we can start to see structures, and I'll come back on how we actually assign different structures to different molecular event. But now with the low magnification tomogram, we are able to place them into the cellular context. For instance, we can see Golgi just next to these ER exit sites. Whereas here at the bottom, we can see an ER exit site that doesn't really have any Golgi anywhere nearby, for example. So here we have a movie, which is a premium of the journal club as opposed to being on a piece of paper. So here you can hopefully see that the tomogram is playing. We have lots of membranes. So now it's gonna play back in a second. So here we have ER membranes. You can recognize them. This one, for example, because they're studded with ribosomes attached to them. This is another one. And there's all these kind of vesicular profiles and clusters coming out of these ER membranes. So this is just actually a technical interjection because before we actually went on and analyzed the molecular details of our tomograms, we improved the alignment by doing the ribosome subtomogram averaging using the WARP-NM routine. So this allowed us to optimize our tilt alignments and get the best possible tomograms. This allowed us also to establish the absolute handedness of our tomogram based on the ribosome structures. So I will skip the details of this technical slide. But eventually we got a very large collection of coated vesicles from our ER exit sites. We ended up with somewhere around 60 tomograms of ER exit sites, from which we have hundreds of coated vesicles. And here, what I'm showing you is a gallery of vesicles that are either completely enclosed or they are budding off the ER membranes and appear to be coated with what we label as the COP2 coat. So here is a picture of an in vitro reconstituted COP2 coat that shows the membrane, the inner layer of the coat here, and the outer coat is much more sparse because it's this kind of thin rod-shaped complexes. So when you kind of get a slice through in a tomogram, you don't see them very well unless you happen to slice it one rod in the longitudinal direction, which is quite rare. And here we have an in vitro reconstituted COP1 coat. So this is the retrograde coat that recycles things back to the ER. And just with our eyes, we were pretty sure that we saw two different types of coats, which were resembling the COP2 and the COP1 coat. This said, when I was showing this to everybody that didn't look at these tomograms a million times like I did, people were looking at me like, I don't really see this. And so we decided to try to do this in a more unbiased and quantitative manner. And we did template matching. So this is a technique where you can take, so we took a structure of the COP2 coat that was deposited in the EMDB, in the EM database, and one of COP1 coat. And with these two structures, we searched around our tomograms to find the positions in our tomograms that gave the best match. And we searched the identical tomograms with these two different structures, and we got a clear preference of the COP1 structures for position in the tomograms that actually we had assigned to the COP1 coat, and for the COP2, two positions in the tomograms that we assigned to the COP2 coat. So that really gave us complete confidence that our assignments were correct. And even though there are some ambiguous positions sometimes, most of them are really clear cut. So at this point, we can definitely say this is COP2, this is COP1. And when we are not sure, we just don't consider that. All right. So this is just a technical slide, technical figure on how we did the template matching. And we basically, what also gave us confidence in the template matching is that even though we are searching in random orientation, all the peak gave us an orientation that is normal to the membrane, indicating that it's finding, a coat that is lying normal to the membrane, indicating it's finding real structure. Whereas if we search, if we look at the peaks, which are much lower cross correlation, but if we still show them in places where there isn't coat, they look like totally randomly oriented. So this was a good way for us to really confirm that we are looking at COP1 and COP2 coats. Right, so now we have a collection of tomograms where we can unambiguously say what is what in terms of coated membranes. And here is a gallery of 9 out of the 60. Of course, we chose the prettiest to put in this figure, but these are really representative of what we normally see. So we also back plotted the ribosomes. And as you know, we can see by eyes that these ER exit sites tend to be ribosome exclusion zones. Then there are these clusters of coated membranes. So we have ER membranes with COP2 buds and COP2 vesicles coming out of them. So here, for example, is a nice bud coming out of the ER membrane, and here are 3 separate individual vesicles. And then we have this vesicular tubular cluster network that is very often coated with COP1 profiles, and much more rarely we see individual separated COP1 vesicles. So here is another movie. It's the same tomogram as before. So again, you can see the ER membranes with, and then these vesicular tubular clusters in the middle with these coated profiles. And now with this movie, I'm showing the results of our membrane segmentation. So we've used this automated AI-based membrane segmentation routines. This is membrane seg. And so here is a 3D view of the membranes. And then the next thing I'm going to show you is the position of the cross-correlation peaks from the template matching. So in purple is the COP1, and in green is the COP2, which allows us to now put pretty colors on our membrane and have a sort of descriptive view of this one particular ER exit site. So you can see there's also some non-coated vesicles. We cannot possibly know what these are. It's likely that they are uncoated events, but of course there's no way to know because cryo-ET is not a dynamic technique. So we're only freezing things at one certain point in time. So we wanted to actually go back to fluorescence to confirm that what we are seeing here is the way that most ER exit sites actually look like, because obviously we only have around 60, whereas there's hundreds per cells. But you know, cryo-ET is not a high throughput technique, or not yet very high throughput. So we did fluorescence imaging of ER exit sites components. So this is 3-color. This is deconvolved confocal microscopy, where we have Sec23 in green, Sec16, which we use as a marker of ER exit sites on the ER membrane, and beta COP, which is one of the COP1 components. And you can see that these 3 proteins are always next to each other at the ER exit sites. They're not perfectly colocalizing, but they're always next to each other. And Sec23 tends to be in the middle. So it looks like what we're seeing is very consistent with Sec23 being somewhere in between the ER membrane and the COP1 coated membranes. Okay. Here we have repeated the same experiment with Sec23, COP1, and ERGIC 53. Now, ERGIC 53 is a marker of the ERGIC compartment, the ER to Golgi intermediate compartment. And again, we see this sort of 3-way localization. We suspected that the COP1 coated vesicular tubular cluster structures we've seen are ERGIC compartments due to the fact that we know the ERGIC is always just opposed to the ER exit sites. And we've done some STED microscopy here just with the COP1 and ERGIC 53, which really shows that the COP1 seems to encircle this ERGIC 53. So this really confirms that what we are seeing is ER exit sites with COP2 vesicles coming out of the ER, and then these ERGIC compartments where COP1 coated vesicles are budding from.
It's so closely opposed to each other. In my conception, it was like there's exit sites, and then there's like the ergic, and then there's like the Golgi, but it's like all there.
Yeah, it's all within a few hundred nanometers.
So
So
yeah, I mean, it's like touching practically. Fascinating. Anyway, sorry, go ahead.
which I think it's why there are all these different models, because in fluorescence you can't really distinguish them as separate entity, and sometimes it looks like they are really connected, uh,
Right.
but they are not physically the same membrane. So
Yeah.
it's that's something that we can only resolve using cryo-ET, which is why we've done this.
Very cool.
Right. So the other thing that we can do now, having all of these beautiful vesicles and buds that we know exactly what they are, we can do some quantification. So we can do some morphometric analysis by fitting spheres and ellipsoids into this three-dimensional reconstruction and counting things. So we can count on average how many COP1 and COP2 membranes are at ER exit sites. We can see that their numbers correlate quite well, depending on the size of the ER exit sites. Interestingly, we see that for COP2, we have many more vesicles than buds, whereas for COP1, we have many more buds than vesicles. And that tells us something about the uncoating, probably, of this. It suggests that COP1 uncoats a lot earlier, upon scission from the donor membrane, that COP2 may be retained on independent vesicles for a little bit longer, because we see a lot more. And we can measure the volume of these vesicles, which for COP2, this is the volume in nanometers cubed, but basically, given that they're almost spherical, it corresponds to vesicles with a diameter of around 70 nanometers. It goes up to 100 or something like that, but we never really see bigger vesicles than that. This is just another figure to show that we see this structure of COP2 from the ER and then the ergic with COP1, whether we are close to Golgi or not. So we have caught some peripheral ER exit sites. These are nowhere near to the Golgi apparatus, which is perinuclear. Here, in fact, we are very close to the actin at the plasma membrane. So this is very far from the Golgi. And we still see ergic with COP1. So somehow these structures will need to be transported to the Golgi, and that's one of the things that, the major things that we still do not really understand in the field. Okay, so a final data figure that I want to share is focusing on a slightly different proteins. Again, it's one of the components of ER exit sites, which is TFG. And TFG has been shown for a long time to be localized at ER exit sites. And this is another one of these proteins that we don't really understand how it assembles and how it works. Scott, you've worked with John on TFG in the past as well. And there are also a number of models out there, one of which suggests that TFG forms some sort of compartments that separate regions for COP1 vesicles from regions for COP2 vesicles. And we do not see TFG in our tomogram. It's probably too small and not regular enough to be recognized and to be seen. But using fluorescent microscopy, we clearly see that TFG seems to be kind of in between. It's together with COP2 at ER exit site, so it doesn't really encircle ER exit site as was suggested before. Right. So in summary, we found that our data supports the classical vesicular transport model. And therefore, on one hand, it tells us that all of the studies that we've done in vitro that led us to learn a lot about the molecular composition and the molecular function of all these code components are actually relevant, which is great. And it also sets a number of questions that have not been resolved. So we have not solved the issue of large cargo transport, because we never see these tunnels that were kind of hailed as the solution for large cargo transport. So maybe these type of tunnels exist in different cell types or in cells that need to transport collagens, for example. but we don't know that. We yet have to find the evidence for that. There's also an issue with cells with high secretory loads. Are these numbers that we see here enough to, for example, secrete thousands of immunoglobulins per second, as our B cells need to do? What happens to the secretory pathway in that case? That's another question that we're really interested in. And also another question is what happens post-ergic. We always see the ergic there, but we know that things need to move to the Golgi. We know they do that probably in a macrotubule dependent manner, but that's another big question that we need to work on. So that's it. I just want to acknowledge again all the authors in this preprint. It's been a great collaboration. It's been great fun. So Katie in my lab, she's been taking on the challenge. When we moved to the creek, it was the first time we had access to a cryofibsem, and we decided to go for not ribosome, but a more difficult project, which was very rewarding in the end. Julia from John's lab did a lot of the, did all of the fluorescence microscopy that you've seen. Andrea from the Francis Crick Institute Structural Biology technology platform, she's amazing. Sander, also in my lab, did some of the initial work on the cryofibsem, and John and myself, of course. And of course, I'd like to acknowledge the funders for the work. All right, that's it.
Thank you. That was excellent.
That was,
Really enjoyed it.
yeah.
I do have some scientific questions before we move on to the second part. Ash, do you have anything first?
No, I'll let you go first.
Okay.
Ask her.
First of all, the tomograms, I don't do this myself yet, although I want to. Um, but in looking at, I've looked at a lot of them before. It's so dense around the vesicles. Like it looks like a lot of protein around the vesicles. Are you comfortable going out on a limb and saying maybe that's TFG?
No.
No. Do you think it's possible?
Um, it will ever be possible, or do you think it's possible with this data?
No, do you think it's possible that that is TFG and that might be why it's so dense
I mean,
around?
TFG is definitely there
Yeah,
because every time we stain by fluorescence, it's there. So it must be somewhere in our images because it's always co-localized with Sec23.
and John has this hypothesis about a cloud of TFG that is sort of surrounding all the vesicles, and I mean, I don't know, it's really compelling thought, at least that maybe this why the reason why it's so dense looking around those vesicles is it's TFG. I know it's maybe too far to be
No,
kind of,
I think it's okay to speculate a little bit, and
yeah,
I think that also it's quite compelling that there's no ribosomes in the area. So that
right.
really tells us that there is some mesh, of other proteins blocking because it doesn't look empty. It looks like there's some dense matrix there, which
Yeah,
could easily be TFG. I mean, I don't see many other candidates, to be honest.
right, right. I mean, the cytosol is very dense, but that looks particularly dense. So anyway, that's
Yes.
interesting. Okay, I do have one more scientific question. Okay, so you have this really interesting sentence in the preprint. You say, it is therefore possible that COP2 transport has adapted and evolved in collagen secreting cells. That's an interesting statement. So is that like you're thinking like maybe coevolution of these of secretion of big stuff and COP2?
So I think, I mean, there's no question that in organisms that secrete collagen, there are factors that localize to ER exit sites interact with COP2 that are not present in other organisms.
Mm.
And it's clear that some of these factors are clearly involved in secretion of collagens. Think about Tango1, think about, even TFG itself has been proposed as possibly aiding collagen secretion. Now, I don't think that the whole COP2 mechanism has evolved a completely different way of doing things, but I think that the way I picture it in my head with this highly dynamic assembly and disassembly, if you can regulate the assembly disassembly kinetics one way or another, you can easily get very different morphologies and very different shapes of the coated vesicles. If you can regulate assembly and disassembly, you can even get vesicles that coat or not, that uncoat or not, before or after they are pinched off the donor membrane. So you can get a lot of room to incorporate large cargo just by regulating how you assemble. I mean, I don't know that this is the way it works, but that's, I guess, what I mean by evolving a system for transport of large cargo is evolving regulators that, will act on the very same molecular mechanisms that we've characterized in yeast and, in vitro for years, but to generate different morphologies.
Right.
Am I making sense?
Yes, yeah, absolutely. Absolutely.
I think that partially addresses the question. I was wondering, it was just beautiful, the pictures and the data and stuff. And I was just like fascinated about how some of these things were excluded. And, particularly in the case like ribosomes were excluded. Why would it create an atmosphere where these can be excluded? This is something that, was boggling me, but, it's very interesting. Although,
This is one of the dangers when you're doing cryotomography in cells is you see so many cool things and you want to investigate them all, but
I
you just don't have the time or you don't even don't know what they are. So
That's a fascinating thing about microscopy, isn't it? Yeah.
yeah.
Cool. Um, hey, shall we move on to the next phase?
Yeah, let's move on to the next part.
Okay, so Julia, we have a few questions for you
Should
about.
I stop sharing my
Yes,
presentation?
yes.
All
There
right.
we go.
So we have a few questions for you about how this paper came together and more broadly your journey as a scientist to reach this point. So I have the first question, and then Scott and I will take turns with you. Okay. So,
Yep.
what was this breakthrough moment for the paper? So there were so many technical advancements I see in the work that you presented, and what was a point where you thought, "Oh, we got to do this, and this is
Yeah.
going to be a story."
It was definitely the first time I opened a 3D reconstruction and I saw what to me looked unambiguously like COP2 coated vesicle. There was actually one in the first tomogram where I've seen it, but it was so clear. So as I mentioned, we had to do all of this kind of funky correlative method, but at the beginning we actually tried to fish in the dark completely. If you count how many ERX sites there are and the volume that you're imaging in one tomogram, you can actually calculate this about, I think you would, by chance, you would get 1 in 100 or something like that tomograms if you just go random. And so we, started collecting data and, nothing until one day I found these COP2 coated vesicles. And that's, when we also realized at the same time that we had to do a correlation to be able to do this efficiently. But by then, I'd seen one, so I knew they existed. So I knew it would work.
I
And
see.
so the rest of the project was actually fun. It was like, happy work because we were going after something I knew was there. So that was, yeah.
Okay, so you've been working in vesicle trafficking for a while. How do you see this particular work in this paper fitting into your overall scientific trajectory?
Right. So I think of myself as a cryo-electron tomography person.
Mm.
So I've done cryo-ET in my PhD when, it was really in its infancy as a technique. And I moved on into the membrane trafficking field as a postdoc because I thought that there were, cryo-ET could really make a difference there, as an up-and-coming technique. And in fact, that has been the case. And now with all these in situ techniques that are really becoming mature and allow us to do more and more with correlation and also in interpretation of the data, there are so many outstanding questions in membrane trafficking that we can finally tackle. That yeah, I really to me, it's like a natural evolution of what I've been doing. Just taking on technical challenges, learning new techniques, which is always fun to find out more and more and dig more and more nerdily into the details of my favorite part of biology.
Yeah, yeah. Um, yeah, I completely agree. I'm so excited about membrane remodeling vesicle trafficking. It's clearly fib milling cryo, cellular cryo-EM is going to be revolutionary for it.
Yeah.
So, okay, so related to this question, like, we're doing a lot of visual kind of stuff. Like, so do you have like an image or a picture or a data point that's like burned into your memory? Is like, this incredible GeoWiz moment?
Yes, I do. And that's back in my postdoc. And that's the time where, so we had reconstituted COP2 budding in vitro, and the first time I actually looked at the tomogram of that, and I could see this very regular lattice that nobody had expected would be there because we knew from your work that the outer coat would form this cage. But I saw this very tight lattice at the inner coat level, and it was so clear that the inner coat had some structural role and some formed a structural scaffold that was absolutely not known before. And that, for me, when I saw that, I was like, wow, what is this? And then we went on and solved the structure of that thing, and we realized it was the inner coat. We could fit all the crystal structures, and so that was probably the surprise from when one image just really told us that there was something completely unknown there that was waiting for us to you know to find out what it was. And in fact, I've got here sorry, it's going out of focus, but I've got your 3D printed inner coat structure, Scott, that you brought when you came to London.
It's great. I'm glad
Yeah.
you still have it.
Yes.
So, Julia, I want to ask you this question. So, which one would you choose as your scientific hero? Cop one or cop two? Oh, and a scientist, and why?
Um, it's gonna be COP 2, not COP 1
Okay,
for sure.
great.
Um, yeah, I thought quite hard about my scientific hero scientist. So I decided to eliminate anybody alive to start with. Just for there's so many inspirational people out there that I know, and I went for quite an obvious choice, maybe, which is Dorothy Hodgkin.
Mm.
I think I'll consider her my scientific hero. Obviously, because she's a goddess in structure determination. I don't need to go into the details of why that is, but also because she made seminal contribution in an environment that was really not, uh, you know, easy. Not at all. So she had health issues, she had family and children, and especially she was working within very antiquated rules that really put breaks in her career because she was a woman. And despite all of this, her passion for solving the scientific puzzle just never let things get in her way, and she got it through. So I think that's how I chose her.
That's a great choice. Yeah,
Yes,
absolutely.
absolutely. All right, last question. So, do you have an experiment but that you'd absolutely love to do, but you haven't been able to either due to time, resources, etc.?
Yes, I do, but I would add a yet to the
Ah,
to the
okay.
question, which is obviously looking at on one hand, looking at cells that are secreting collagen. I'm
Ah,
dying
yeah, that's
to
what I
I'm
want to
dying to
do
see how that
too.
actually happens.
Yeah,
But on the other hand, I would really, really like to optimize the in vitro reconstitution with a human system to be able to reconstitute, from native membranes, collagen things, because I mean, I care about the morphology, but I really want to know the molecular details of how these cages are assembled and regulated. And for that, the in vitro system is great. We can really get high resolution, and I really, really would like to be able to work on that.
absolutely. All right. Well, Julia, thank you so much for joining us. Really enjoyed your presentation. We really appreciate you taking your time to tell us about your science. And that does it for this episode. So we hope that you will all join us again for the next episode of Structure Club.